Hi, class. So I am going to start lecture one for unit one for statistical methods. So I'm going to share my screen to the PowerPoint. We're going to get started. Let me make it big. Okay, so this is the Psych 2317 first lecture. So I'm just going to start by saying why we need statistics as psychology majors, right? I touched on it for like a second in your syllabus video. But why are we doing this? Well, because behavioral scientists do research. Statistics are a way that we can describe what we did have quantitatively, right? What we were using words for, we need a way to be able to apply numbers to that so that we can describe the numbers that way. So then that helps us see why statistics is important. Because I know as a psychology major, I was like, no, I just want to help people. Like, I don't want to do research. I'm not going to do research. I just want to do counseling or social work or human resources, maybe. Maybe you don't ever intend to do anything further in the psychology world than your bachelor's. But psychologists are human researchers. And they're scientists and behavioral scientists. And so scientists do research in all of the statements that we make and the claims that we make, we're able to say these things because we have data on our side. We have research, right? We've done some sort of experiment, and then we've taken those results and we've described them quantitatively, and we've done some computations, and then we have these numbers that are supporting what we're saying, not just, oh yeah, I saw a change. I saw something different. And I was curious about it, right? Um, so this well, first let's do an example. So you can see here, we're going to name the color of each word as quickly as possible. So I'm going to just read the words, right? Blue, green, red. Wow, that was very easy, right? I was able to do that very easily. So now let me try the second post-it note over here. I'm going to try to read the words. Blue, green, red. Well, if you had a stopwatch and you were timing me and you were, you get down to like the milliseconds and you could get it really specific, you would be able to see that I was slower while reading these. Over here, you can see that blue is written in blue ink, green's written in green ink, and red is written in red ink. But on the second note, blue is written in green ink, green is written in red ink, and red is written in blue ink. So if I tried to do this quickly as possible and I had this longer list, right, it would be difficult and I would be a lot slower when the words were incongruent or when the words and the colors did not match up. So let's say these are our two conditions, right? In the first example, the first condition was easy and the second condition was not as simple. So a behavioral scientist might look at that and go, I wonder why that happened. Why was the second condition slower? Because something curious is going on there, right? Well, behavioral scientists have actually been looking at that for a while. It's called the Stroop effect, and it's been around since the 1930s, but it's very interesting. And it causes a cognitive psychologist to pause and go, this is something about how what we read is automatically processed. So as a scientist, I want to know what's going on, right? We know that we're slower at naming the color when the color and the word are incongruent, don't match, presumably because we automatically process words when viewing them, right? We're presuming that we haven't been able to prove that quantitatively, right? We don't have any numbers to support that. We just saw that the second one was slower, and that's what we're assuming. To understand the Stroop effect and other aspects of human behavior, we conduct research to test our competing models. But what does competing models mean? So that's the statistics part, right? That's the statistics portion of what we're learning, right? These competing models. So that's why we need statistics. We need it because we, as psychologists, are observing all of these things all the time, all these phenomena that we think are interesting, right? Are different. We're observing helping behaviors. And if people want to stop on the side of the road to help change someone's tire, or bullying in schools, or results of medication on children's ADHD behavior, anything, right? Anything that we're studying memory, and if this smell has an effect on memory improvement, or any gamut of items that psychologists want to study in human behavior. We need statistics to be able to tell us if we actually found anything or not. So when I'm talking about um, research, right, we're talking about how it consists of setting up the experiment, measuring those things, and then collecting that data back. And then the model, that's the quantitative instantiation of some behavior. So it's our best guess, right? It's us turning the Stroop effect, something that we can describe with words, into something quantitative that we can describe with numbers. So we're going to make two models. We could set it up like this. So we could say that the Stroop effect, this model one, has no difference, right? So there is no Stroop effect at all. There's no difference reading the congruent and the non-congruent words. There was no difference. Um, so saying that there's a difference in time, whether there's no difference in time or there is a difference in time, that time difference, present or not, is our number, right? This gives some numbers to what we're trying to describe as the Stroop effect. So we might say model number one equals 0. No Stroop effect, no difference. And maybe model two says there is a difference and it's positive, right? So here we're seeing there is a Stroop effect and it is more than 0. That just gave numbers to literally any definition or any guess that we could have made about what was happening when we read these congruent and non-congruent words, right? We just broke every single phenomenon that could have happened into there was no difference or there was a difference. So we will spend the whole course figuring out what that looks like and what that means and how we actually do that. So the primary question we're always going to be asking is, now we've broken it down into two models. Which one of those models best observes the data that we have? So how do we answer that question? How do we figure out which model is more accurate? Which one is the best description of it? Well, we have to be able to describe our data. So in Unit 1, we'll talk about that, right? What's the most typical types of measurements and how do we expect these measurements to vary? What's the standardized effect size? We'll talk about effect size in Unit 2. And then we'll make inferences with our data. So we'll take that data and then we will infer things from that. We'll use tools of probability that we'll look at in Unit 3, and we'll make best guesses about the true magnitude of our effect while accounting for uncertainty in our measurement, which we'll talk about in Unit 4. And then in 5 and 6, we'll talk about estimating the size of the effects that we have, because effect size affects our data, right? And then we'll look at the adequacy of our models, right? Did we choose models that are adequate for what we're doing, right? Is it a t-test? Do we need an ANOVA, some analysis of variance? Should we use Bayesian? And so those are covered in Unit 6 or 8, and then chapters 9 and 10, or Units 9 and 10 of your textbook. And so we'll have to figure out, well, which model should we be using? Like, what's the most adequate and the most appropriate for this situation? And then we'll also review basic statistical concepts that I think hopefully you're familiar with from other stats courses, but if not, this is the place to become familiar with them. I think, sorry, I'm trying to decide if I want that to be a separate video or not. Let me unpause the screen sharing. Okay, I decided to keep it as one lecture. Okay, so let me share the screen back again. Sorry about that. Okay, reviewing some basic statistical concepts. Population. So the population is the group of individuals that are the focus of the experiment. So maybe we're looking at a specific clinical population, individuals diagnosed with depression, or specific student population, all the third graders at this elementary school, or all of the third graders in America or college students as a whole, right? Some population that we want to focus on and find out about. So the sample is the subset of the population that we use in our experiments, right? You cannot possibly go do an experiment on every third grader in America or every college student or even every freshman at Tarleton State, right? We need to get a sample from that group. And so that's the difference between our population and sample. Statistics is the science of inferring something meaningful about the population when all you have is the sample. So some properties we can use to describe our data set are center and variability. So when we're talking about describing center, we're talking about measures of central tendency. So mean, median, mode. Those are the things I think you've probably heard in other courses, right? You probably know what the mean, median, and mode is. And then the other one is our types of variables. So we have interval/ratio variables, and that's the difference in equally spread values at one part of the scale. The same difference is equally spread across values at another part of the scale. So, for example, the difference between 32 degrees and 40 degrees is the same difference as 62 to 80, right? 8 degrees is the same, right? There's an 8-degree difference, whether we're talking about that first part or that second part. So the spacing is the same and it's meaningful. Ordinal is meaningful, but the difference is not necessarily equivalent. For example, high school, bachelor's, master's, PhD. So there's some difference in these values and it matters, right? But the difference between 12th grade and high school and a bachelor's degree in college is not the same, right? You took 12 years to get to the end of high school, but it only takes 4 years-ish to get to the end of college. And so those differences are not equivalent. And then nominal means there's no order to the categories, right? It could just be, they're like word categories, right? Brown eyes, blue eyes, green eyes, hazel eyes. There's no order to that, right? Brown eyes aren't better than green eyes, and green eyes aren't better than hazel eyes. And so there's no order to it, but they are differences in categories. One other thing that I wanted to point out is in ratio. No, no, just kidding. I don't want to confuse you. We'll talk about that later. So some measures of central tendency. The first one is mean, right? The sum of all the values in a data set divided by the number of observations, right? We only use that in interval ratio data. And so this is mean in statistics, right? We call it X bar. And so you can see this X with a bar on top of it. That is what the symbol for mean is in our psychology statistics. And so you would add up all of those data set. So maybe there's five, and you add up all of those five different scores that you have, and then you divide it by five because that's how many scores you had, and that's your mean. So we had 2, 3, 5, 8, and 12. So we added it up. 2 plus 3 plus 5 plus 8 plus 12 equals 30. And our n was 5. We had 1, 2, 3, 4, 5 examples. So 30 divided by n equals 6. So our mean equals 6. X bar equals 6. And so here's the formula for that, which looks kind of scary, but it's not. So this is saying the sum of all of the x1s divided by n, divided by your number. So if we had these five data sets to like shortcut it, we're just going to call this our x's, right? So we had x1, x2, x3, x4, and x5. And so we took the sum of all of our x's, our xi's, and then we divided it by n, which was 5, right? 1, 2, 3, 4, 5. And that gave us our mean of 6. Easy peasy. And then we have the median. So that's the middle number, which we only use for ordinal or interval or ratio. So we put the values in numerical order. If there's an odd number of values, it's easy, right? The median is that middle number. So if we had 2, 3, 5, 8, and 12, 5's in the middle when we put them in order, numerical order, and that's easy. But if there's even, then what do? There's no middle number. Well, we take the average of the two middle numbers. So we put them in order, and then we go, okay, these are the two middle numbers, 5 and 6. 5 plus 6 divided by 2 equals 5.5. So there's our median. And then mode is the most frequently occurring value. So let's look at that same data set, right? So we can use it for nominal, ordinal, or interval ratio. We can use it for any of them. So if our survey results said that 40% of residents in a city have cats, 30% have dogs, 10% have birds, and 20% of those, our sample have no pets. The most frequently occurring, the mode is cats, um, because 40% had cats, so they had the most. The mean and median are meaningless with nominal data, right? We can't, what do we do with that? That's just nominal, right? We ask people to tell us if you were a cat owner or a dog owner, but we can't group that, right? Cat owners aren't better than dog owners. Dog owners aren't better than cat owners. So we can't get mean and median, but we can get mode. Go to this one. So we have, hey, what's your voting preference? Republican, we had 375 people. Democrat, 510 people. Independent, 141 people. Declined to state, 84 people. So find the mode for that, right? Which one is most often occurring? Whatever level of measurement, right? There was nominal, ordinal, and interval ratio. So mode, we could use mode or median. We could use mode, median, and mean in the interval ratios. So why might we choose to use the median as a measure of central tendency instead of the mean? The mean is really sensitive to outliers, so really extreme values, which might result in skewed data. The median is robust or more resistant to outliers or really extreme scores. So for example, we have 2, 3, 5, 8, 12, and 400. So our mean would be all of those numbers, right? Xi's added up and then divided by our n, 1, 2, 3, 4, 5, 6, so we have an n of 6 equals 71.67. The median is our two middle numbers, so 5 and 8 divided by 2, 6.5. So the mean is 71.67. That's a huge number. What does that mean? Well, the median is 6.5. What does that mean? Right? So the median would live kind of here, right? In between 5 and 8. Well, the mean is all the way over here, right? Between 12 and 400. So which measure would you say is the most representative? So if we did the mean, median, mode of these income levels, our mean score is $46,000, median is 16,000, and mode is 14,000. Which one do you feel like most represents this population here? Good answer. Positively skewed distribution. So sometimes if we have extreme high scores, the right hand tail is longer than the left hand tail. And so you know which side's the tail if you make a fist and you see how this kind of follows the curve of your bell curve and then it's skewed. And then we have the other skew. I know it's backwards in the camera, but you can use your fist to kind of see which way it's skewed. Extreme low scores result in the left tail being longer than the right. Skews it negatively. And then here's our normal distribution, right? A nice, beautiful little bell curve that only has a little bit of tail on each side, but no skewness. So measuring variability, the spread or dispersion, it refers to how spread out the data are, by how much, on average, each data point differs from the mean. We care about that because the larger variability means these scores are more spread out. And the deviation score is the difference between each individual measurement and the mean. So deviation is the distance, right? And we want to standardize that deviation. We want to have standard deviations. So maybe we were looking at these fish, right? So the mean length was 3 inches. And so the length of the fish in inches, so we have 0 to 6 inches. And so if the mean is 3 here in the middle, then we need to see how much all of these fish deviated from the mean. So we've got some negative deviations, we've got some positive deviations, and we need to see what those deviations looked like. So here's the deviations, actually the numerical values for here. So for our -1, 0, +2, +1, -2. So here are those numbers. And so here's what the scores were, right? These XIs, 2, 3, 5, 8, 12. Um, this is a different example, not the fish example, but another example. The fish was just to kind of let you see it visually. So here's our scores, 2, 3, 5, 8, and 12, our data. And so we need to find the deviation. So we need to look at that data minus the mean, right? Minus X bar. So if the mean was um, 2, so we're going to subtract 4. We're going to subtract 3. We're going to subtract 1. We're going to add 2, add 6, positive, negative numbers. And so then we've got our xi's minus X bar, so minus our mean squared. So then we'll square them all. So 4 squared, 16, 3 squared, 9, 1 squared is 1, 2 squared is 4, and 6 squared is 36. And we did this because we needed to get rid of these negative numbers, right? So by squaring a number, we get rid of the negatives. So we found the average, right? So now we add these up. So we have 16, 9, 1, 4, and 36 divided by 5, 13.2. So this is our average squared deviation. Because when we did this, the average deviation was 0, which was not helpful at all. So our mean here was 6. And when we took 2 minus 6, we got negative 4. We added all this up, right? Our deviation was 0. That's why we squared it. And because look, negative 4 plus negative 3 plus negative 1 is 4, 5, 6, 7, negative 8. Or, yeah. And then 6 plus 2 is positive 8. Well, negative 8 and positive 8 cancel out each other, and that's nothing, so not helpful. But when we squared it and we got rid of these negatives, we were able to find the average square deviation that way, which is helpful. So variance is the average square deviations. So this thing that we just did is your variance formula. So you might want to remember that. So we have the sum of all the xi's minus the mean squared divided by the overall number. So for this data set, 2, 3, 5, 8, 12, we found a mean of 6 and a variance of 13.2. Variance is supposed to measure the average distance from the mean, but that does seem too big, right? Doesn't 13.2 seem like that's pretty big difference from the mean of 6? I mean, that's like over double. Like, that seems interesting. But since we squared the deviations, our average is squared. So our variance, that's difficult to interpret. What do we do with that? So what we do is now we take the square root of our variance. So if we take the square root of what we just got of our variance, that equals our standard deviation, right? Not our average standard or average deviation. This is our standard deviation. So for our data set, 2, 3, 5, 8, and 12, standard deviation equals the square root of the variance. So if our variance was 13.2, our standard deviation is 3.63. Well, that's a lot smaller. That makes a lot more sense, right? So on average, the individual data points deviate from the mean by 3.63. So you can see if there's less variance, we have the scores closer together, right? Our data closer together. But the more variance there is, the more these scores stretch out, or the more our data set stretches out. So easy peasy, beginning lecture. Make sure you understand all of those terms really well. Maybe be making a cheat sheet of those formulas so you can reference those throughout the semester. And yeah, just go from there. Email me with any questions.